Assalamu alaikum, dear students. I am Muhammad Rauf, lecturer, Department of Zoology Cast. Welcome to the course Principle in Animal Life 2. This is lecture number 22. The topic is how DNA was designated as genetic material. Discussing agenda for the topic will be historical background. DNA is the transforming factor in bacteria. Avery, MacLeod, and McCarty identified the transforming substance as DNA. DNA is the genetic material in certain viruses. Lecture outcome. After watching and listening this lecture, students will know about the historical background of how DNA was identified experimentally as the genetic material of living organisms. Historical background. Swiss physician and biochemist Friedrich Mischer in 1860s isolated nuclei from white blood cells in pus on soiled bandages. He isolated white blood cells from discarded hospital bandages, and then he isolated an unusual substance in the nuclei of those white blood cells. He discovered in the nuclei an unusual acidic substance containing nitrogen and phosphorus. He and others found it in cells from a variety of sources. Because the material resided in cells' nuclei, Mischer called it nuclein in an 1871 paper. Subsequently, it was called a nucleic acid. Walter Sutton in 1902 proposed chromosomal theory of inheritance along with Theodore Boveri. In that theory, he postulated that the behavior of chromosomes during meiosis is similar to the behavior of Mendelian hereditary factors. T. H. Morgan was a famous Drosophilist. He was an opponent of both Mendel's work and chromosomal theory of inheritance, but he himself experimentally proved that the genes determining eye color in Drosophila are located on X chromosome. So after his findings, he became one of the followers of Mendelian genetics and chromosomal theory of inheritance. He in his experiments identified genes to be present on chromosomes. Now, chromosomes initially misled researchers regarding the biochemical activity of the genetic material. When the composition of chromosomes was studied, it was revealed that chromosomes are composed of DNA plus associated proteins. So then a new debate started that whether DNA is the genetic material or proteins are the genetic material. DNA is composed of only 4 types of nucleotides, whereas proteins are the most diverse group of macromolecules. There are hundreds of thousands of different types of proteins identified and properties studied. So more people were of the opinion that proteins could be the genetic material of living organisms. DNA is the transforming factor in bacteria. So once the chemical composition of chromosome was identified, then a new debate started to experimentally prove whether DNA is the genetic material or proteins are the genetic material. So the first person to investigate in this area was British medical officer named Frederick Griffith. He was trying to develop a vaccine against pneumonia. Pneumonia is caused by a bacterium called Streptococcus pneumoniae. Griffith had two strains of the bacterium. One was smooth, pathogenic, disease-causing, virulent. The second strain was non-pathogenic and it was rough. So Griffith was surprised to find that when he killed the pathogenic bacteria with heat and then mixed the cell remains with living bacteria of the non-pathogenic strain, some of the living cells become pathogenic. Furthermore, this newly acquired trait of pathogenicity was inherited by all the descendants of the transformed bacteria. Apparently, some chemical component of the dead bacteria pathogenic cells caused this heritable change, although the identity of the substance was not known. Griffith called the phenomena transformation, now defined as a change in genotype and phenotype due to the assimilation of external DNA by a cell. So this figure shows various experiments performed by Frederick Griffith. In the first experiment, he injected living smooth cells which were pathogenic into the mice. The mice died as expected. Then he injected living rough cells which were non-pathogenic and they didn't had any effect on the mice and the mice survived. In the third experiment, he killed the smooth pathogenic cells with heat and then injected them into the mice. The mice remained healthy because the pathogenic bacteria were already killed by the heat. Now the fourth experiment was something which resulted in lot of discussion amongst researchers. In the fourth experiment, he mixed the living rough cells with heat killed smooth cells. He mixed those two types of cells and then injected them into the mice. Now if you can see here, none of them when injected individually, resulted in the death of the mice. So Frederick Griffith was expecting that the mice will survive, but to his surprise, the mice died. And when he studied the blood of these mice, living, smooth strain bacteria were thriving inside. So it was something of great surprise, not only for him but for the people around it that time. Now, what had happened inside the mice? Griffith called the phenomena transformation, now defined as a change in genotype and phenotype due to the assimilation of external DNA by a cell. This type of permanent genetic change in which the properties of one strain of dead cells are conferred on a different strain of living cells is called transformation. So he hypothesized that something from the heat-killed smooth bacteria has have transferred to the live rough strain and changed or transformed the live rough strain into a smooth pathogenic one. Scientists hypothesized that some chemical substance, a transforming principle or factor, was transferred from the dead bacteria to the living cells and caused the transformation, caused the change. Now, after this experiment, there was another debate amongst the researchers that what has transformed the live, rough, non-pathogenic bacteria into smooth strain. What has actually transferred from the dead pathogenic strain into the live non-pathogenic strain and changed it and changed its properties, whether it is a protein or a DNA or fats or carbohydrates. So these three researchers then individually isolated different macromolecules from the smooth strand, and in a different sets of experiments they investigated the transforming substance. In 1944, Avery and his colleagues MacLeod and McCarty chemically identified Griffith's transforming factor as DNA. They did so through a series of careful experiments in which they lysed split open smooth cells. They split open the smooth cells and then they isolated lipids, proteins, polysaccharides, and nucleic acid from those lysed cells. They tested each fraction to see if it could transform living rough cells into smooth cells. The experiments using lipids, polysaccharides, and proteins did not cause transformation. However, when Avery treated living rough cells with nucleic acid extracted from smooth cells. The rough cells were transformed into smooth cells. So they first isolated different fractions of the smooth cell bacteria, and then they tested each fragment for its property of transformation. The transformation was only observed when nucleic acid was used. A nucleic acid which was derived from smooth cells, when was mixed in a solution containing rough cells, it transformed the rough living cells into the smooth living strands. Although today scientists consider these results to be the first definitive demonstration that DNA is the genetic material, not all scientists of the time were convinced. They are talking about this particular experiment. This was a very solid proof that DNA is the genetic material, but still many people were not accepting that DNA can be regarded as the genetic material of all living organisms, because the experiment was performed with bacteria, which is a simple prokaryote, and there are hundreds of thousands of species of living organisms. So the other researchers wanted further evidences in other organisms to nominate DNA as the genetic material of living organisms. They were not satisfied with the findings only in bacteria. Many thought that the findings might apply only to bacteria and might not have any relevance for the genetics of eukaryotes, because bacteria is a simple prokaryote. So the findings in a simple prokaryote cannot be applied to diverse eukaryotic organisms. During the next few years, new evidence accumulated that the haploid nuclei of pollen grains and gametes, such as sperm, contain only half the amount of DNA. You already know that a diploid cell has 2n chromosome number and a haploid cell has n chromosome number. And it was the findings of the other researchers in germ cells that the germ cells contains half the amount of the DNA as that of the somatic cells. Because scientists generally accepted that genes are located on chromosomes, these findings correlating DNA content with chromosome number provided strong circumstantial evidence for DNA importance in eukaryotic inheritance. So here what they want to say is that the scientists already knew that the genetic material lies there within the chromosomes, whether it's the DNA or the proteins, and they further knew that the haploid germ cells contain half uh, the DNA content when compared with that of a diploid cell. Now, it was the third experiment which provided further evidence to the DNA as the genetic material. This experiment was performed with a bacteriophage, Bacteriophages are viruses that infect bacteria. The experiment was performed in 1952 by geneticists Alfred Hershey and Maratha Chase. So bacteriophages were used in that experiment. Bacteriophage word means bacteria eaters. These are the viruses that infect bacteria. Viruses are much simpler than cells. On the right side, you can see this illustration of a virus. This is the head region, which is a protein coat, and inside that protein coat lies the DNA or the genetic material of the virus. Now, this virus attacks bacteria, but it does not enter into bacteria, means this protein coat lies outside, it remains outside, and only the DNA, the genetic material of the virus, enters into the bacterial cell. To produce more viruses, a virus must infect a cell and take over the cell's metabolic machinery. So once the viral DNA enters into the bacterial cell, it hijacks the bacterial DNA and the replication machinery of bacteria and other assets of bacteria, and it uses all those resources within the bacterial cell for its own advantage, for making more phages. Phages have been widely used as a tool by researchers in molecular genetics. Viruses are also used extensively in biotechnology. They act as vectors in gene therapy. So we continue with the previous topic. In 1952, Alfred Hershey and Maratha Chase performed experiments showing that DNA is the genetic material of a phage known as T2. This is one of many phages that infect E. coli, a bacterium that normally lives in the intestine of mammals and is a model organism for molecular biologists. Model organisms are the ones which are used by researchers to find some specific phenomena or to study the function of a gene, and then they apply those findings to other living organisms. At the time, biologists already knew that T2, like many other phages, was composed almost entirely of DNA and proteins. They also knew that the T2 phage could quickly turn on E. coli cells into a T2 producing factory that released many copies of new phage when the cell ruptured. Some T2 could reprogram its host cell to produce more viruses. You must have studied the two types of life cycles of bacteriophages. One of them is lytic life cycle, which is also called host and guest relationship. The other one is lysogenic, which is called the first one is called the lytic, which is the host, which is the master and slave relationship. The other one is the lysogenic, which is the host and guest relationship. But which viral component, protein or DNA, was responsible, means which viral component enters into the bacterial cell and hijacks the replication machinery of the bacterial cell and use it for its own advantage. Hershey and Chase answered this question by devising an experiment showing that only one of the two components of T2 phage actually enters the E. coli cell during infection. They used radioactive isotopes, as already mentioned, that the bacteriophage consists of an outer coat of proteins, and proteins are composed of cysteine amino acids which contains sulfur. On the other hand, DNA has phosphate group, so they used two types of radioactive isotopes. With one type of radioactive isotope, they labeled sulfur to take proteins, and they grow that particular culture separately from the other one, because proteins but not DNA contain sulfur. Radioactive sulfur atoms are incorporated only into the protein of the phage. In a similar way, the atoms of radioactive phosphorus labeled only the DNA, because phosphorus is only present in DNA, not the protein, because nearly all the phages phosphorus is in its DNA. In the experiment, separate samples of non-radioactive E. coli cells were infected with the protein-labeled and DNA-labeled batches of T2. The researchers then tested the two samples shortly after the onset of infection to see which type of molecule, protein or DNA, had entered the bacterial cells and would therefore be capable of reprogramming them. So that was the experiments performed by them. They labeled some bacteria with radioactive sulfur, and in other batch some they labeled some viruses with radioactive sulfur. Sulfur is there in the proteins, and protein coat resides on the outside and houses the DNA. So they grew that culture separately from another culture where they used radioactive phosphorus which is present in the DNA. Then they took non-radioactive E. coli cells and added them to both the batches. They allowed the bacteriophages in the two batches to infect the E. coli cells, and after some time they isolated this medium and used a blender to free phage parts outside the bacteria from the cells. When the bacteriophages infected the bacteria, after some time they agitated the mixture and they separated the bacteria from the viral particles, and after centrifugation there were two layers formed. The lower layer was a dense one which contained bacterial cells, because bacterial cells are heavy, so they settled at the bottom, and the phage particles, the protein coats, they are lighter, so they settled in the upper lighter region. And here the same was done here. The heavy bacterial cells settled at the bottom, whereas the lighter phage particles were in the upper region, transparent region. Then they checked for the radioactivity, because here they had already incorporated radioactive sulfur and radioactive phosphorus. So at the end, they checked these solution for radioactivity. In that particular solution, radioactivity was observed in the upper lighter region, which consists of only phage particles, whereas in the second batch, the radioactivity was observed in the denser region where the bacterial cells were present. So after this experiment, it was concluded that the phage particles, which are composed of proteins, they stay outside, and the DNA of the phage particles enters into bacteria and hijack the cellular machinery of bacteria. So it means the instructions for the synthesis of more phage particles is present in DNA. So DNA is the genetic material of bacteriophages. Thank you very much. Allah Hafiz.